Adenine is the only nucleobase that does not have a carbonyl group on it, so there's no double bond to an oxygen on the base here. You can see that it forms two hydrogen bonds with its complementary nucleotide. And then there's the nucleoside is either adenosine or deoxyadenosine if it's missing the oxygen on the 2' carbon here. And adenine is named after the Greek word for gland, in reference to the pancreas. And you can see Greek here, similar to the prefix adenine. So here's adenosine, the nucleoside, with the ribose sugar here. And then here's adenosine triphosphate, so it's the nucleoside with 3 phosphate ions on it. And I would assume that each one is -1 charge. You can see the minus on the oxygens here. Then the gamma phosphate here is showing a hydrogen on one of the hydroxyl groups. So there's still a -1 charge on that gamma phosphate. And this is ATP, which is, the energy usually comes from like glucose or something, and then it kind of just gets stored as ATP, which is kind of like the universal energy unit used by the cell. So here's ATP. You can see it here on the right side. You've got your adenine base. And here's where the base is going to form hydrogen bonds with this hydrogen here and this negative polarized nitrogen. And then here's the ribose sugar and the 3 phosphates. And as we just saw, these hydrogens are probably missing, and then either one of these is going to be potentially having 1 hydrogen and 1 negative charge. And then here's the oxygen that would be missing for, here's the oxygen that would be missing for a deoxy version of this nucleotide that would be used for DNA construction. Here's 1', 2', 3', 4', 5'. So 5' is where the phosphate is going to be attached, and then the 3' is where the growing chain will be growing. And then the base numbering, I'm not sure if it's important, but the number 9 node here is what it attaches to the ribose. So this is number 9. And then number 1 and 6 are where the hydrogen bonds are going to be formed. Here's just AMP, adenosine monophosphate, kind of just being rotated in space here. So you can see this is where the hydrogen bonds would be forming to connect. And then here's the ribose, and here's your 5' carbon and your 3' carbon. And this is the RNA version with the oxygen here. And then there's also the cyclic adenosine monophosphate, which has the phosphate forming a bond with that 3' carbon's hydroxyl group to give it this cyclic component here, and that's a second messenger. So there'll be some first messenger that's probably extracellular, and then that first messenger signals attaches to some signaling protein and generates these cyclic AMP second messengers. Adenosine can also be methylated. So here's N6-methyl adenosine. You can see the methyl group is added to that nitrogen. This is the 6th node here. Sixth node nitrogen. So 6N is where this N6 comes from, and that's where the methyl is added. So this is a way to tag an adenine base in DNA or RNA to just regulate gene expression or any other reason that the cell would need to keep track of something. And you can see here in mismatch repair in bacteria, there's this GATC sequence, and the adenine is methylated in that as a way to let the bacteria know which strand of DNA is new and which strand is old. So the old strand will have the methylation on. You can see this is the same both directions. There'd be a G, A, T, C. So I can add a methyl group to each strand, and then when it duplicates the DNA, the old strands will have the methyl groups and the new strands won't have it, and so the repair process can know which strands are old and which strands are new and correct the new strands to match the old strands. And I think this is only in bacteria. So you can just see here adenine forming hydrogen bonds with, in this case, thymine and DNA.